Hello everyone and welcome back to Biology with Mrs. Evans. Today we are going to continue our conversation on cell transport, but before we do that, we need to talk about concentration. Concentration is simply the mass of a given solute in a given volume of water. So what that means is let's say you have a solution that says 10% salt. That means that 10% of the volume is going to be salt and 90% of the volume would be water or whatever other solvent was used. So we need to talk about concentration because concentration is vital to the movement of particles. So in order for particles to move, there has to be what we call a concentration gradient, which is a difference in concentrations. So in elementary school, you learned about diffusion, and diffusion simply is the movement of particles from an area of high concentration to an area of low concentration. The particles are moving on their own without any input of energy because they are moving from high concentration to low concentration until equilibrium is reached. In the water lab, I talked about nature, and I said that nature likes for things to be in balance, and you got to see that in the what I called the wick trick. Remember, one glass was completely full of water, another glass was completely empty, and all I did was use a paper towel to bridge the two cups, and you saw water move from the full cup into the empty cup until the water levels were equal, until equilibrium was reached. And that's exactly what happens in diffusion. So in the water down a rope video, you saw the guy add food coloring to the water, and this is just to review diffusion real quick. Area of high concentration, so here's where food coloring is initially added, so that's going to be area of high concentration. And again, the particles are going to move without any input of energy, so they're starting to spread out until equilibrium is reached, where everything basically is evenly distributed throughout. And notice that in the video that we watched, you didn't see the guy steer it or have to do anything to those particles in order to get those particles to move. And because there is no input of energy of any kind, we call diffusion a type of passive transport. Passive means no input of energy. So what are some examples of things that can move directly through the phospholipid layer? That would be carbon dioxide and oxygen. Remember when we talked about the cell membrane, we said that small particles or particles that do not have a charge can move directly through. And the reason why they have to be uncharged is because the phospholipid layer, the lipid portion of it is nonpolar, so it's only going to allow other nonpolar substances to move through. And remember, the little space in between the phospholipids are relatively small, so they have to be small in order to fit those little gaps in between the phospholipids. So carbon dioxide and oxygen can move directly through the phospholipid layer, and again, they're moving from an area of high concentration to an area of low concentration, so there is no input of energy in order for those particles to move. Facilitated diffusion is another type of passive transport. Again, passive meaning no input of energy. In facilitated diffusion, if you look at the term facilitate, it means to assist. So when we talked about the structure of the cell membrane, we said that a particle that was too big to go through the cell membrane, the phospholipid portion of the cell membrane, would need to go through a protein. Or if the particle carried a charge, it would need to go through a protein. And two examples of substances that move through the cell membrane in this way would be glucose. Glucose is too big to move through the phospholipids, and then water is another molecule that would need to go through a protein, because remember, water is a polar molecule, so it does carry a charge, so it cannot go through the phospholipid layer, so it must go through a protein in order to get through the cell. So facilitated diffusion means that the particles are moving through a protein. The protein is what's giving the assistance. And again, it is passive, so the particles are still moving from an area of high concentration to an area of low concentration. In middle school, you learn the term osmosis. Osmosis is simply the movement of water across a selectively permeable membrane. again, the water is going to move from an area of high concentration to an area of low concentration. So if you look at the notes that I have on Blackboard, you will see this particular diagram and you'll see this in motion, so you'll get to see how it works. But for this, just know that here we have an area of high concentration for sugar, and over here we have an area of low concentration. Now, sometimes it's easier, requires less energy on the cell or input to move water than it would be to move these bigger molecules. So oftentimes the water is going to move as opposed to the larger molecules, or it could be the larger molecules simply will not fit through the cell membrane, so water is going to be the one that is moving in order to equal out the concentrations. So what happens is that this is an area of high concentration, this is an area of low concentration as far as we talk about sugar concentration. So what happens, water is going to move from this side over to this side, and it's going to do that because it wants to equal out the concentrations. So water is going to move over here to dilute, which means basically to weaken the concentration of sugar so that the two concentrations will eventually become equal. So in the slides that are on Blackboard, you'll see that this level over here is going to get lower as the water moves over to this side, and this side the water is going to move to a higher level until equilibrium is reached, when the concentrations are equal. So know that osmosis is a type of facilitated diffusion, and it is simply the movement of water across a selectively permeable membrane. Water balance. Maintaining water balance is crucial to the cell, and in our discussion there are some terms that you need to know. One is hypotonic, the other is hypertonic, and then the last one is isotonic. Hypotonic, this guy right here, means that one solution's concentration is lower than another when you're comparing the two. So if you have a 10% salt solution, it is going to be a hypotonic solution compared to a solution that has a 20% salt concentration. And then the opposite of that would be hypertonic solution. Hyper means a higher concentration of solute compared to another solution. And the way that I remember that is, just remember, if somebody says you were hyper, that usually means that you ate a lot of sugar probably. So a hypertonic solution has a higher solute, in this case sugar. And then the last one is isotonic. Isotonic means equal concentrations. So what happens to cells that are placed in the different types of solutions? You're going to get to see this in action when we do the gummy bear lab. So let's look at what happens. So remember, a lot of times it's easier to move water than it would be to move these larger salt sugar molecules. So what happens if you place a cell in a hypotonic solution, what it means is that this concentration outside the cell is lower than the concentration inside the cell. So let's say it is 10% on the outside and 20% on the inside of the cell. What happens is water is going to move from the outside of the cell into the cell. And the reason, again, is because water is moving into the cell to try to dilute the concentration that is higher in the cell. So inside the cell it would be hypertonic, outside the cell would be hypotonic. If you place a cell in a hypertonic solution, so now the outside is going to have a higher concentration compared to the inside, so the inside would be hypotonic, then water is going to move out of the cell. Again, water is moving out to try to dilute the concentration and bring them into equilibrium. So the cell is going to shrink. So when a cell is placed in a hypotonic solution, so a lower concentration, the cell is going to swell because water is moving into the cell to try to dilute it, to try to dilute the concentration inside the cell. When it's placed in a hypertonic solution, meaning the higher concentration is outside the cell, water is going to move out of the cell to try to dilute the hypertonic solution that the cell is placed in. So what happens to the cell? The cell is going to shrink. And then isotonic solution ISO, remember, means equal. So water is going to move in and out of the cell in basically a steady pace. There's not going to be a net movement of water, so there's not going to be any change in the cell size. So water's moving in at the same rate as water moving out, so there is no net change in the cell, so the cell will remain the same. So far we've talked about transport that does not require energy. Remember, that term for that is passive transport. So you're moving from an area of high concentration to an area of low concentration. And again, the term for that is down a concentration gradient. Just like water moves down a hill because there's a change in elevation. There's no need to pump it. It's just simply moving from an area of high elevation to low elevation. So there are opportunities, or there are needs, I should say, of the cell to move things against the concentration gradient. So instead of going from high concentration to low concentration, particles are going to need to move from an area of low concentration to an area of high concentration. And we call that active transport. We call it active transport because the cell is going to need to use energy in order to move those particles against a concentration gradient. Just like if you want to get water up a hill, you're going to have to use a pump. All right, you're providing an energy source to get the water to move up the hill against an elevation gradient. So cells, there are going to be times when they need to move molecules against a concentration gradient. An example of that is what we call the sodium-potassium pump. Inside the cell basically wants to have very low sodium content. So they're going to actively pump the sodium out of the cell. And inside the cell, they want to have a higher potassium content. So they're going to be pumping potassium into the cell. So in order to achieve that balance that it needs, it's going to be moving ions against the concentration gradient. And that is important because that's how our nerve impulses work. They have to maintain this delicate balance of difference in charges so that nerve impulses can basically move down the neuron cells in our body. So in order to do that, the cell is going to use energy, and the energy molecule that is going to be used by the cell is ATP, and we're going to talk about that in Unit 5 in detail. So just know for right now, active transport is the movement of particles against a concentration gradient. It's going from low concentration to high concentration. Again, the cell is going to be using energy, and proteins are also going to be helping with those movement of particles against that concentration gradient. Okay, so when we talked about the cell membrane, I also said that sometimes the particles are too big even to pass through the protein in the cell membrane. And when we did the water lab, you guys got to see hydrophobic sand, and I said that sometimes the cell membrane would wrap around these larger particles in order to move those particles into the cell. And you guys got to push on the hydrophobic sand and see how it wrapped around your finger. And that's exactly how the cell membrane works. So if the particle is too big, or it's trying to move, let's say, a bulk of items into the cell, then the cell membrane is simply going to wrap around the particle. So it wants to bring in these particles here. So notice that the cell membrane is starting to create an envelope around these particles, and it will completely pinch off, and it forms what we call a vesicle, and it's going to transport those materials into the cell. Because it's moving particles into the cell, it's called endocytosis, so it's moving large particles into the cell, and then exocytosis is basically moving particles out of the cell membrane. So the process is basically in reverse. So now you have a vesicle that is moving the particles out. It will fuse to the cell membrane and basically open up and then empty those contents of the vesicle to the outside. So endocytosis, moving in large particles into the cell membrane or into the cell, sorry, and then exocytosis, moving large particles out of the cell. There are two types of endocytosis that you need to know. One is what we call phagocytosis, and that is bringing in basically solids into the cell, and we refer to that as the cell is eating. You'll see that in the worksheet that you guys have. And then the other type of endocytosis is penocytosis, and that is basically bringing in liquids into the cell. And the way that I remember that is I like to drink virgin pina coladas. Pina colada is a liquid, so penocytosis is bringing in liquids into the cell, whereas phagocytosis is bringing in solids. So this is just showing you again how that works. Endocytosis, it wants to bring in these larger molecules that cannot pass through the protein of the cell membrane. So the cell membrane is going to basically start to create this envelope, and eventually it will pinch off, forming a vesicle that is going to bring those materials into the cell. Here's an example of an organism that uses this, and that would be an amoeba. So you notice that this would be a food particle that it wants to basically eat. So you'll notice that the cell membrane is starting to wrap around. It will eventually completely close, creating a vesicle, bringing in that food to the cell. Contractile vacuoles. So what that is a vacuole is basically a storage device inside the cell that allows for storage of water, food, waste, and things like that. So contractile vacuole is a vacuole that has the ability to contract. So when it is full, it's full of liquid, and if this was a cell that wanted to move, it would squeeze the little contractile vacuole and it would expel the water out this way, and the cell would move basically in the opposite direction, just like an octopus and a squid, how they swim. They squirt the water out, and it propels them forward. So contractile vacuole has the ability to contract and move the single-celled organism. Alright, so let's review. How can you determine the type of transport? That is a big part of this unit. Given different pictures, you have to determine what's going on. And the best way to do that is to use a 3-step process. This is what I use myself, and that is first to see how the particles are moving. So are they going from high to low concentration, or are they moving from low to high? So I have the pictures here. I apologize for the blurriness. This is the best that I can find from Google. So notice here, these guys are going from high concentration to low concentration. So I know that that is a mode of passive transport. Then the second thing would be is once you determine how they're moving, is to determine basically how are those particles getting into the cell. So these guys right here, they're going directly through the phospholipid layer. So this is an example of diffusion. The second way, remember, is for the particles to go through a protein. And notice it's going from high concentration to low concentration, and it's going through a protein. So that is facilitated diffusion. And then the third step would be to determine what is being moved. So a lot of times they'll say this would be, let's say, sodium. And this would be, well, not sodium because it wouldn't be going directly through the phospholipid. So let's say this is oxygen. So it's going through. And then this would be, this one we'll say is potassium. It's going through the protein. So again, it's going through a protein, and it's not water, so I know it's not osmosis. So look at what's going through, because if it was water, let's say this is water, then this would be osmosis. So look and see, how's it getting into the cell? What's the concentration difference? So if I see this one over here, on the right here, notice that there are 2 molecules here, and the arrow is pointing this way. So it's going from an area of low concentration to an area of high concentration. So I know that that is active transport. And because it's going from low to high, notice that you see the input of energy, allowing for those molecules to move against a concentration gradient. So you may want to pause the video and write down the step to help you determine the type of transport. So first look to see how's it moving. If it's going from high to low, then you know it's some type of diffusion. Then you need to determine how's it getting into the cell. Is it showing it going through a protein? If it is, then it's facilitated diffusion. If it's not going through a protein and it's moving from high to low concentration, that is simple diffusion. If it is going from low concentration to high concentration, you know that that is active transport. And sometimes you'll see pictures of the cell membrane completely wrapping around materials, moving that in, and that would be endocytosis. Again, it is physically wrapping around those substances, so it also uses energy in that type of movement as well.